So good morning, buenos dias. My name is Paul Diehl. I am the Chief Operating Officer at Cellecta, and welcome to the symposium. I appreciate the few of you that were able to get up at 8 o'clock in the morning and come out and hear some of the talks that we have, which I think will be pretty interesting, so I think it was worth the effort, I hope. But before we get started on that, I just want to give you kind of a quick overview of the company and introduce the speakers. And so Cellecta was founded about 16 years ago now. We are a small company south of San Francisco in California, in Mountain View. And the company is a technology-focused company. We have about 25 people. Most of them actually do work in the lab. And the company itself is not venture funded. It really started on the basis of SBIR grants and used that to develop a couple technology platforms, which I'll mention briefly, and then some of our speakers will go into more detail. The focus really is on technology for understanding the genes and genetic elements that are driving biology. So the genes driving disease progression or differentiation, or that would make good targets for various drugs, et cetera. And the focus is to develop assays or tools that can do this in a very massively parallel way, because it's a complicated question to understand how all of these genetic elements function together in order to make biology. And the main tool that we use for that is next generation sequencing. And the NGS technology over the last 10 to 15 years has gotten much more accessible, and it gives you the ability to really deconvolute sort of complicated assays and look at and pick apart the different elements that are creating change. And we try to make the assays accessible to ordinary laboratories. So they don't require large-scale automation, LIMS systems, et cetera. These are assays that regular laboratories experienced in molecular biology or cell biology can carry out. We've had some success over the years. We have over 400 publications on our website that have cited different products and services that we offer. And this is a searchable database, so if you're interested in any particular application, you can actually go on the website and take a look at that and try and search to identify publications or studies that might be related to what you're interested in doing. The technology platforms that we have developed and offer products and services for fall into three broad categories. The CRISPR RNAi genetic screening, and this is using large pooled libraries that express either sgRNA or shRNA, and the company actually started using shRNA technology, and then of course as CRISPR came on the scene, then we started to we focused on developing libraries for that too. And we provide the libraries. We can do full-scale screening, and we have individual constructs, and we have various support products for the pipeline here for screening. And the talks today will deal with the CRISPR platform, and also to some extent single-cell technology. We have barcode libraries which are different than the CRISPR libraries because they're expressing unique sequences that can be introduced into cell populations. And these then can be used to label cells and look at different subclonal populations within a cell culture to identify ones that are maybe sensitive to particular drugs or changes, et cetera. And these can be used also in combination with sgRNA depending on how you design the libraries. And then we also have a multiplex PCR expression profiling system called the driver-map assay, and this is used as an alternative to RNA sequencing. It's much more straightforward. It's it's an RT-PCR platform, and it's a multiplex RT-PCR, so you can amplify targets from all protein coding genes in the human genome in a single reaction. And then our experience with lentiviral technology and also cell engineering. We all offer a number of products and services for that. We do have an exhibit booth downstairs, so if any of you are interested in hearing more about any of these technologies, please stop by the booth and we'd be happy to talk to you. And so our two speakers today, our first one is from Cellecta, Dr. Donato Tedesco, and he's going to talk about a study that he did looking at 10x genomics, single-cell profiling, and comparing that with FACS-sorted profiling, and looking at the expression of after introducing an sgRNA library, looking at the transcriptional response with TNF-alpha in the cells and seeing how the results compare with the two different approaches. And then after Donato finishes, we have Dr. Matteo Marzi, and he's from the Istituto Italiano of Technology in Milan. And he's looking at also a perturb-seq type of system where we have a CRISPR library, in this case CRISPR-I library, targeting long non-coding RNA and looking at a single-cell basis and seeing how the long non-coding RNA, or identifying how the long non-coding RNA is important in breast cancer cells. And so with that, I'd like to introduce Dr. Tedesco.